HPLC, that is high performance liquid chromatography, is the analytical technique most widely used in the pharmaceutical industry for the testing of the pharmaceuticals. It is not only used for the products, but it is also used for the raw materials and many others. And it is very important to understand the HPLC technique basics. From long time I was thinking to make the videos on analytical instruments also. So now I have started, and one by one I will be making videos on HPLC instruments, like then analytical instruments, HPLC techniques, and I will prepare the videos on analysis of the pharmaceuticals. Now coming to the introduction to the HPLC, as it is a high performance liquid chromatography, as the name indicates, it is used for the analysis of the pharmaceuticals. HPLC is the most advanced analytical technique for the separation, identification and quantification. The basic principle of HPLC is the chromatographic technique for the separation of the analytes, and then it identifies and then it quantifies. Separation occurs in the HPLC because of the interactions between the mobile phase and the stationary phase. If you are aware about the chromatography, in chromatography there are moving phases and the stationary phases. So one is the mobile phase, it is liquid solvent that carries the sample, and stationary phase is the solid material or adsorbent in the column, and these phases interacts with the compounds. Based on the interaction, the analytes get eluted from the column by the flow of or through the use of mobile phase. If there is a strong interaction between the analyte and the stationary phase, then there will be a slower movement. If there is a weak interaction, then it will be a faster movement. So based on the slower movement and faster movement only, the chromatography techniques are developed. HPLC technique is one of them, and HPLC technique uses the stationary material or stationary phase present in the column, and each compound elutes at a unique retention time. So if you are separating and analyzing the analyte, that may be the pure API, that may be the API in the formulation like tablets, capsules and many more. Then it may be the separation of the analyte, that is drug, and the impurities. All these materials or analytes have the specific interaction with the stationary phase, and this interaction depends not only on the stationary phase but also on the mobile phase. So development of the mobile phase, selection of the stationary phase, and optimization of the process parameters of HPLC are the main points to be learned and the points to be studied. Each compound has a specific interaction, and that's why based on that specific interaction it gets eluted from the column, and that time is called as the retention time. Coming to the main components of the HPLC system, so it has reservoirs, it has pumps, injector, column, detector and the data system. Reservoirs hold the mobile phase. Mobile phase may be made up of the different concentrations of the water, methanol, acetonitrile. Sometimes OPA is also used, orthophosphoric acid, to maintain the pH. So different combinations are there to make the mobile phase based on the polarity, based on the chemical nature of the analyte. Pump delivers the mobile phase under the high pressure. An injector introduces the sample into the system. Column is containing the stationary phase. Column is packed with the stationary phase for the separation. and column is the heart of the HPLC system. Detector identifies the compounds. Different detectors are there present in the HPLC system, and these are selected based on the characteristics of the analyte. So these are UV detectors, PDA detectors, DAD detectors, then fluorescence detectors are there based on the chemical nature of the analyte. Data system is there to record the signals detected by the detectors, and it generates the chromatogram. You will get the retention time and you will get the area under the curve, and based on that you can quantify the materials. There are different types of HPLC like normal phase HPLC, reverse phase HPLC, ion exchange HPLC, size exclusion HPLC. So these are some of the types. In normal phase HPLC, polar stationary phase is there and the mobile phase is non-polar. These terminologies we will understand in the upcoming videos. But for now, you understand that normal phase means polar stationary phase, and reverse phase is the non-polar stationary phase like C18 column, and the mobile phase is polar in nature. So it is most commonly used in the reverse phase. Normal phase, polar stationary phase, reverse phase, non-polar stationary phase. And based on this polarity of the stationary phase, polarity of the mobile phase, and polarity of the analyte, chemical nature of the analyte, the HPLC system works and the method gets developed. Ion exchange HPLC works by separation of the analyte by their charge interactions, and size exclusion, that is gel filtration, in this separation is by the molecular size. Applications of the HPLC, so it is widely used in the pharma, food, environmental analysis, clinical and medical. In the pharmaceuticals, it is used for testing the drugs, excipients, then performing the stability studies, doing the assays, the dissolutions, and assessing the impurities. So many uses are there. So HPLC system is most widely used in the pharma industry, and it is precise, high speed and sensitive technique for the analysis. It separates, identifies and quantifies the compounds in the mixtures, and in the pharma it is mostly used. For example, if you want to analyze the API or if you want to analyze the impurities, then HPLC is the most important technique. This is a summary of the HPLC system. In upcoming videos, we are going to learn the deep understanding about HPLC. Thank you for watching the video.